Gene anatomy. What is gene? A piece of DNA, or it can be piece of RNA, that contains the primary sequence to produce a functional biological gene product. Gene product can be in the form of protein or it can be in the form of RNA. In other words, we can say that entire nucleic acid sequence necessary for the synthesis of a functional polypeptide, protein, or it can be RNA. Genetic information is stored in the DNA. Segments of DNA that encode protein or other functional products are called as genes. Gene sequences are transcribed into RNA, which is called as messenger RNA. So genes, they are the functional units which are present on the chromosomes. Genes can be in the form of DNA genes, which actually produce polypeptides, or we can say that polypeptide producing genes, and genes can be RNA genes which produces RNA. Messenger RNA is translated into proteins that perform most of the life functions. So DNA is converted into RNA and RNA is translated into the proteins, and proteins, they perform life functions. If we see that what is the anatomy of a gene consists of three main components. One is called as, or the first one is called as open reading frame. Frame means from start codon up to stop codon. Start codon, that can be ATG, and the stop codons, they can be TGA, TAA, and TAG. So all the entire sequence which is present between the start codon and the stop codon is called as open reading frame. So that is the first component of the gene. The second main component of the gene is upstream region with binding sites. In other words, we can say that the Tata box, GC box, and cat box, which are present towards the upstream region of the gene. This is the second main component of the gene. And the third main component of the gene is poly A tail, which is present at the three end of the gene. So these are three main components of the gene: open reading frame, upstream sequences, and the poly A tail. This is the gene anatomy of a typical prokaryotic gene, in which we can see that this is the open reading frame that starts with the start codon, and here is the start codon ATG, and the open reading frame ends with the stop codon, and the stop codon is TAG. So these are, and in addition to open reading frame that starts with start codon and that ends with stop codon, there is a region which is called as promoter region, where there is Tata box. So this is the structure of a typical prokaryotic gene, in which we see that there is a open reading frame, there is a start codon, there is a stop codon, and there are promoter regions. This is the gene anatomy of the eukaryotic gene, because there is a little bit difference between the anatomy of the prokaryotic gene and the anatomy of the eukaryotic gene. Eukaryotic genes, they are usually complex in nature, which consist of exons and introns. Here we can see that this is the promoter region of the gene. These are the 5 UTR regions. Then there are introns and exons. This is intron, this is exon, again intron, exon, intron, exon. So in case of eukaryotic genes, we see that the genes consist of exons and introns. This is the 5 UTR region, And this is the promoter region. 5-UTR region consists of exon and intron. If we see that at this region there is start codon and there are codons in case of exons and there is donor site. So this region has been expanded or it has been magnified, where we can see that there is a start codon and there are different codons, and then there is donor site, mean end of the exon. Here is the region that is combination connection between the intron and the next exon. Here we can see that again there is acceptor site. In case of the boundary of intron and exon, that is AG and CC, or it can be in case of donor site, it can be AG and GT, and in case of acceptor site, it will end with AG. So these are the exons. And if we come at the end of the gene, we can see that here is stop codon, TGA. Here is the stop codon, and this sequence is called as 3-UTR region. And at the end of the 3-UTR region, there is poly A tail, AATA. So this is the poly A tail. If we do the comparison between the prokaryotic gene and the eukaryotic gene, prokaryotic gene is simple. There are no introns, while in case of eukaryotic gene, there are exons and introns. This is the comparison between single exon gene and multiple exon gene. Here we can see that the gene consists of a single exon, a start codon, then there is a stop codon, and gene consists of only single exon. While in case of multiple exon genes, we can see that exon, intron. So there are more than one exons in case of multiple exon genes. So in case of eukaryotes, there can be genes which consist of single exons, and there can be genes which consist of more than one exons. So if we conclude that what is gene? Gene is a piece of DNA or a piece of RNA that contains the primary sequence to produce a functional biological gene product. Gene product can be in the form of polypeptide or it can be in the form of RNA.